Hello everyone, I'm Álvaro Tavares, and on this presentation I will explain some of the details on optics that are on the base of microscopy, and I will try to simultaneously give you some details of the history of microscopy and how microscopes were developed, in this case just visible light microscopy. Before jumping into the details of microscopy, and I'm sure you're all aware of this, but I would like just to remind you that the way we see that allows us to see the world and everything that matters to us, we are completely dependent on this optical system that it's our eye, the human eye, and the light will pass through the pupil and it will be focused by the lens on the retina. That is where the image will be formed. And when the light rays that are emitted from that come from the object that we observe, and the way the lens work, and we're not going into the details, they will focus on the retina, but the image will be seen upside down. For the purpose of our talk, of this talk, this is not really important, but when building a microscope, and if we want to create an image that truly reflects the position of the object being seen, this can be one important detail. And of course, as I'm aware, you all know that the light that allows us to see, the rays of light, they behave like waves, and such detail is represented in here. So light has a wave-like behavior, and there's the visible spectrum. We can only see waves, let us say, that range from around 7 nanometers to 400 nanometers wavelengths. And so that means that shorter wavelengths rays, we actually cannot see them, or longer wavelength rays, we don't either see them either. They can be detected with the proper apparatus, but our eyes can only distinguish, are only sensible. Our detector is only sensible, the retina, to waves with these wavelengths. And this leads me to tell you about some of the properties of light that are important for to understand what I want to say on the next slides. Of course, you're all aware of some of the properties of light, like the three that are represented in this cartoon. So an incident light on any object can be absorbed fully by the object, and there's no light going through. Can pass through the object in case the object is transparent to that wavelength, so it's not opaque, it will be transparent to some wavelengths, or the light can be reflected. In fact, this is an important detail, because the reflection of light over the object is what allows us to see colors in the different objects. So basically, if all the light was reflected by an object, we would see the object as being white. If everything was absorbed by the object, all the wavelengths, we would see it as black. And white light, so light from the sun, as you also aware, let us say, if it has the 7 colors of the rainbow, when the incident light hits an object, in this case a leaf, we see, as our perception of the object, the color of the object is the wavelengths that is reflected. So in this case, this leaf would absorb all or most of the rays except the green. It's the green that it's being reflected, so the observer would have the notion of this object as being green. One important property of light for the purposes of our talk is the phenomenon that we call refraction. So refraction of light basically consists on the change in direction that the light suffers when moves from a medium to another medium where it moves at a different speed. So as an example, the light reflected from this fish, it moves through water, and when it reaches the boundary between water and air, it will be refracted at a certain angle. So the fisherman will have the perception that the fish is at this position, while in fact the real position of the fish is a different one. So this is a phenomenon that we observe every day, more often than we care to think. For example, if you look into a glass of water with a straw inside, it will look as if the straw is broken at some point at the boundary, when in fact we all know that it's not the truth. So the refraction happens only when at the boundary. It will be determined, the angle that the direction, the angle created from the movement of light is dependent on the wavelengths. It's dependent on the direction the light hits the surface, and we can actually see a good example in here where white light is coming incident on this prism, and it's refracted in a certain direction, and then it again finds another border and it changes again on the opposite direction. So this is an important detail that I will be referring to through the rest of my talk, which is the refraction of light. A consequence of the refraction of light, and that the refraction differs to different rays according to the wavelengths of these rays, is if we have a mixture of rays of different wavelengths, like in white light, that are constituted with the different colors of the spectrum that we can see, when this complex mixture of colors hits the surface of the crystal, so each ray will be refracted to a different angle, and it will be refracted again when it hits the second surface, so when it changes from hair to glass and again from glass to hair. So there will be a refraction, and we can increase this side, and of course if we do so, we can disperse, we can separate the different colors that constitute the original ray of light. And we observe this usually also when you look through glasses and with water, or in nature. The rainbow is nothing more than the dispersion of light caused by drops of water that are in the air. So these characteristics of light, in particular refraction, is in a sense what makes a lens work. And as you can see in here, if we have an object that we want to observe, the light that is transmitted, that moves in the direction of the eye, when it reaches a lens, and this is a simple lens that we will talk about different types of lenses in the future. So light is refracted, and it will reach, in fact, a second lens, the lens of the eye, and it will be focused on the retina of the observer. But because of this refraction of light, there's a phenomena where the image is in fact amplified, because the image that forms in here, it's again as if the light ray was coming from this position and from this position. So the image that the eye is forming in the retina is in fact a virtual image of this side. So there was some degree of amplification of the image. This will depend on the, again, on the size of the lens and the distance that we have between the lens and the object. But basically, this is the function of a retina taking into consideration of a lens taking into consideration these properties of light I just mentioned you about. So taking these properties into consideration, the refraction of the light rays when they move from one medium to another medium, and the angle of the surface, we can have the double convex lenses, that's the ones we've been talking about, but in fact many other types of lenses can be made that will diverge light, that will disperse light in different ways, and according to their own wavelengths, this dispersion can be higher or lower. So we can have lenses this way, that also that they are only convecting one side and plane on another side. They can be concave, and so each one of these lenses will allow us to do different diversions, let us say, to the light rays. And how can all this be used for us to build a microscope? In fact, people have always been fascinated by the properties of translucent crystals and glass since ancient times, and they learned how to use these properties of the crystals to amplify images, especially very small objects that they wanted to observe. So the use of reading stones, as it says, it's quite old. It's even before Christ. So people would use small pieces of glass or crystals that they would find to help them, in this case more advanced, to read. And later on, when the manufacture of glass was perfection, people would use what we call reading stones that can amplify quite well the letters of a text to be used. And they were in use up until quite late, until we were capable of doing proper lenses or magnifying glasses to read. The use of those stones were also adapted to create reading lenses. So reading lenses have been used ever since man knows glass, but the first person to actually describe the characteristic how to make a convex lens and using it for the building up of a reading lens was Ibn al-Ata in 1021, so an Arab, in the Book of Optics, the first book really dedicated to optics, and influenced enormously the construction of nice lenses to be used in reading lenses. Only later on, with the use of better construction of glass, people were able of doing spectacles that would help people to read. They were notoriously difficult to keep in the face. Only much later on technology allowed to do the spectacles that we now use, but spectacles, the way they are represented on this figure, were first developed in Italy around the 13th century, and they used in the beginning quartz because the optical glass was not yet very good. So for professional reasons, many people needed to look very closely to objects like tissue makers that were making fabrics, like in Holland. They really needed to observe up and close the tissues being manufactured to observe the quality. But this is not so easily done because our eye has severe limitations. Objects that are very close to the eye cannot have their images brought to focus on the retina. The accepted minimal conventional viewing distance is around 10 inches or 25 centimeters. So these lenses were being used, the primitive lenses were being used to help to see these fabrics. So it's not a surprise that in Holland, eyeglass makers started to play around with lenses to help the merchants to observe the tissues they were manufacturing. And in fact, Zacharias Janssen in 1590 were the first ones. This is disputable, but apparently they were the first ones to manufacture the first compound microscope. So the difference between a compound microscope and a simple microscope is the compound has more than one lens. It has multiple lenses inside the tube. And they were capable of doing the first compound microscopes that a replica is in here in this picture, and they could actually amplify images up to 3 times when it was fully closed, or they could be elongated, and at that stage it would amplify an image up to 10 times. It's interesting to notice that the way a compound microscope function is by successive amplification. So you can see here the objective lens will amplify the image of the object, and it will create a virtual image inside the compound microscope. And then the eyepiece, or the ocular, as we nowadays call it, will focus this virtual image into the eye of the observer. So the observer will have the notion that it's looking at a much larger image, while in fact the object is small. So in fact it will help amplify. So the compound microscope is based on this principle, but it suffers from a series of problems to work that we will discuss in a bit. Nevertheless, it is interesting to notice that the principle of the compound microscope and of the telescope is basically the same. What it changes is the focal distance from the object. And like Galileo was interested in developing telescopes, he was also very much involved in developing the microscope. So there's a lot of similarities between the two objects. And in fact, the way light is reflected and refracted inside is in a lot of ways very, very similar. So the first records we have of observation of use of a compound microscope for scientific purposes are from Marcello Malpighi, an Italian professor that was in fact considered the father of embryology and histology. So he described many structures of the human body and the animals, and he also observed wood, and he was an extremely curious person, making observations and using an extremely simple compound microscope. So these compound microscopes are difficult to focus on the sample, and in particular because the sample is difficult to illuminate. So most samples were not transparent, and the light had to be to reach the sample either using glasses, candles, and it was one of the major problems. Nevertheless, using such a compound, Malpighi was capable of describing in detail many anatomical structures, and in here are some images of one of his publishings. The lungs, you can see the structure of the lungs and the representation of the tissue, and in particular you can see the detail of the capillaries, the structure of capillaries, and what we nowadays know it will be cells. Also making use of these simple compound microscopes, another giant in microscopy is Robert Hooke, who was later the president of the Royal Society in London, and using also simple compound microscopes, and you can see the system to illuminate the sample. He made many observations. He observed insects, and he described many structures, but he really became famous because he coined the term cell, and it was published in his famous book called Microscopy. And in that book you could read that when he was observing slices of cork, that he noticed that everything was perforated and porous, and those pores he called cells, a term that stayed up until today. Nevertheless, science hasn't really caught up the use of microscopes up until van Leeuwenhoek described his own work. So Leeuwenhoek is incorrectly called the inventor of the microscope when he created this simple microscope that we see on the image on the right. So there were already many others using compound microscopes or even simple microscope. But Leeuwenhoek was very creative, using a microscope to observe samples that are positioned in here, and he was very careful describing his own observations. And with a microscope of like this, he could attain a magnification of about 275 times, and he submitted his work for publishing in the Royal Society of time with Robert Hooke. So the earliest microscopes that were really used to describe cells, among them are the Leeuwenhoek, that has this very simple structure. So this is a concave lens in here, a convex lens, sorry, in here, and the sample would be positioned right at the tip of this needle, and then two different screws you can see in here, this one and this one, will allow the user to position the sample more into focus or less into focus with this microscope. Not only that, Leeuwenhoek, because he was very curious, he dedicated his time to see samples of water from different pools, from different tissues. So he was the one who really described bacteria, and he found out that in a simple drop of water there was a lot of living organisms, microorganisms that we can see. He also observed the blood cells. He also described the sperm cells. So he was elected, because of all of these discoveries, a full member of the Royal Society in 1680. And as you can see from these images, and in particular from this one, you can see that this is not a really simple microscope to use. It's not so simple to do observations and to then draw cartoons out of them. Nevertheless, they have a great advantage over the compound microscope of the time, which was they were easy to position according to the source of light. So you could use more intense light, more or less intense light, and this was good enough for these early observations. And because of these observations, there was an enormous interest in this microscopic world, and it was at this time then many people decided to start, okay, this is something worth studying, and a lot of other researchers started to try to develop a good microscope. And in here we can see another good example of the observations registered by van Leeuwenhoek in the year of 1717. In this case, spinal cord of a cow described with great detail. And the observations of Leeuwenhoek then really were a big success, let us say, at the time, and many other people decided to start trying to use microscopes to make observations on anatomical structure, or start using them to observe smaller things. In particular, what were these small animals that were described in the water pools. Nevertheless, there were severe limitations to these microscopes, either the simple ones, either the compound microscopes. There were several problems, mainly because the quality of the lenses were not that good. And in fact, one of the major problems was what we cannot see here in the image of this horse. It's the chromatic aberration. So chromatic aberration is derived from the fraction of light that we've seen a few slides back, and it's the dispersion of the colors that depends on wavelengths of the light ray and of the medium being traversed. So maybe we can see this better on the next slide. So as we spoke before, refraction of light is the change in direction that light suffers when changes from a medium to another medium where its speed is different. So traveling through air, then through glass, and then through air again. Every time there's a frontier between the two mediums, the light changes its direction. And this direction, the angle that it suffers, the angle that implemented is due to the angle of the surface itself being traversed. It depends on the wavelength of the ray that it's incident, and so when we have light coming through one of lens, that one of the sides it's plane and the other one is convex, what you can see is each ray can be refracted in a slightly different angle. So they're not all refracted the same way. So instead of focusing, all the rays are not focusing on the same spot. So this is the focus spot of a perfect lens. If the lens is perfectly well made, it's perfectly made, all the rays will focus on one area, and in this case, this is the most common effect. The different rays, they don't all focus on the same spot. So this, and every single ray of this is the same wavelength. So this is what we call spherical aberration, and it needs to be corrected for us to have a clear image. Different aberration from this that I just described, the spherical aberration, is what we call chromatic aberration, because these rays can be all blue rays. So we will have the dispersion of rays instead of focusing in one spot. They dispersed, but imagine that we have three different. The incident light was composed of three different wavelengths. So the blue, the green, and the red. So each single one of these rays will have each one a spherical aberration. So all the colors will suffer an aberration different from the other ones. So this is what we call chromatic aberration, and it can be corrected. So the way to correct this was in fact discovered by Chester Hall in the year of 1730s, when he made the observation that newly made glass or flint glass dispersed the colors in a way different from old glass. It was a characteristic of the glass. So he tried it out to make different lenses, and he designed a system that uses a concave lens and a convex lens close to each other in a way that we can focus the incident light, the incident rays, on a single spot, on the focus point. So this is a correction, it's a way to correct chromatic aberration. But while Chester Hall had an empirical solution, so he tried out different types of glasses and curvatures to make his lens to correct this, the chromatic aberration, Joseph Lister solve the problem of spherical aberration using pure mathematics. So he published his discovery in 1830, and this has allowed the construction of lenses that are capable of correcting the spherical aberration that we were just mentioning. So this allowed the manufacturers to design lenses already prepared to correct spherical aberration. The advances in the quality of lenses and the microscopes that were being used allowed many people to start working on this new field of science, which is to analyze this life, small organisms that were living in drops of water in every pond, river, that people could see, and it was unimaginable before. So CDO in the year of 1878 was the first one to create the word microbe, to use the word microbe as referring to these small organisms, that basically means in ancient Greek microbios, it means the short-lived, and they have such an importance in our lives as we know nowadays. It almost sounds like that everything that had to be defined in optical microscopy was made in the 19th century. But in fact, the two other giants in microscopy, and Ernest Abbe and Carl Zeiss, a name that is familiar to everyone, they did seminal work and in the construction of microscopes and in defining some of the rules to use microscopes. But perhaps I should start by saying that Abbe, the law of Abbe, determine one important factor, which is what's the maximum resolving distance that we can attain on a microscope. So resolving 2 points is what's the minimal distance between two objects, that when I'm observing them on a microscope, what's the minimal distance that I can distinguishing them as two different objects. And Abbe was capable of saying that it's directly the distance between the 2 points, the distance between the 2 points is directly proportional of the wavelength that we're using. So the smaller the wavelength of the incident light, the smaller the distance, the 2 points that I can see and resolve under the microscope. To better explain what I'm trying to say, it's better to show this as an example. So two objects that are close, like these two objects, I can see them, I can distinguish them as separate entities only if they have a certain if they are a certain distance apart. As we approach them one to another, at some point we will have the perception that we cannot say if this is just one object or two objects or three objects that are so close that I cannot distinguish them. So the incident light that I use to observe these objects is what the wavelengths of that light is what will allow me to separate, to distinguish as separate objects that if they are really close. So the Abbe's law basically established a limit to what it's possible for me to observe, because we know the wavelength of the minimum light ray that I can see with my own eyes. So below that I will not be capable of resolving two objects under a light microscope. And why is that? Why is there a limit to the resolving power? What's the physics behind all this? So the physics is, there's another characteristic of light that we haven't mentioned before yet, which is the light diffracts around corners. So when we make light waves of light pass through one small aperture, a hole in this wall, light will bend around this corner and around this corner. So the larger the opening it is, the less we observe this effect. But this is the characteristic that allows us, if this was a sound wave and we were sitting in here, we would still be capable of listening to the sound, because the wave of sound will bend around the corner. So this is the phenomenon that happens. So obviously this diffraction depends on the wavelengths, and it depends on the size of this hole that this aperture that we have in here. It's present in our everyday light. All waves suffer this phenomena, even large waves, as you can see in this aerial picture of sand in a harbor, where you can see these barriers that prevent the water waves coming, and the effect is easily observed on the sand caused by these waves. So what are the implications? The implications is, as the wave passes through an opening, like in here, if this could be one lens and light coming through the lens, so when the light bends around the corners of the lens, it will diffract and will create a pattern, a diffraction pattern. And to the observer, this diffraction pattern is seen in here as small light circles. Of course, as I told you before, the different wavelengths, so blue and green, they will diffract differently. So as you can see, not only we see circles of light, but you also see some chromatic aberration in these circles. And the first person to describe the reason of these circles of light was George Airy in the year of 1835, and the presence of these circles of light limits enormously the resolving power of a microscope, because the circles of light will superimpose. So you can see two different objects perfectly resolved, and you can see these two are not resolved, so they are below the resolution of the microscope. And these circles of light, now usually we refer to them as Airy disks, due to the name of George Airy, which was the first person. So just to summarize, so our the capacity of seeing two objects as separated instead of one single object, this is what we call our resolving power of our microscope, or the resolving power of our eyes. And according to the Abbe's law, he said, according to law, we will never be capable of separating 2 points if they are so close that the distance is less than half of the wavelength of the light that we're using to see the object. So this is the maximum or the minimum distance between two objects that allows us to separate them. So one way that we can use to try to minimize this effect is, you can see in this image again, where I'm just showing you again the refraction that light suffers when it passes from one medium to another medium. If we are looking to an object that is covered by a cover slip, and this is the lens of our microscope, so when light passes through our object to be seen, when it reaches the glass, the light can be refracted. When it passes from the glass to air, it will be refracted again. When it passes from air to the first glass in the lens, it will be refracted again, and then when it comes out of the glass, it's refracted again. So this every time light changes, this will cause problems in our image. So ideally, we wouldn't have the difference in this medium. So light would ideally would pass all these mediums. They would have the same refractive index, let us say. And one way to solve this problem is, instead of having between our cover slip, our sample, and the objective lens of the microscope, we can use an oil that has a refractive index similar to this glass and to this glass. So with the appropriate choice of an oil, because the refractive index of all these three layers is the same, the light will go through them without changing direction. And this would increase the quality of our image. And the importance of choosing an oil with a refractive index adequate is perfectly demonstrated in this movie, where you can see that this oil that has the refractive index of the glass cup inside, it will make it appear as if nothing is in there. So basically there's no contrast. The light is not refracted when it changes from the medium, in this case the oil, to the glass. So the light will continue its path without changing direction, and without changing direction it looks as if there's nothing in there, as if it's fully transparent and fully the medium is just one. So the choice of an appropriate oil when looking into sample in optical microscopy is a crucial detail that it's often not taken into consideration. So Abbe and Zeiss were obviously aware of the importance of the oil, and they developed an oil immersion system in order to take into account these refractive characteristics of light. So they developed oils that match the refractive index of the glass that were used to make the lenses and the slides and the cover slips. And with such a system, the first Zeiss microscopes, the 150 years ago almost, with the lenses with a maximum of 1.4 numerical aperture, it could be, the system would allow us to resolve 2 points that were distanced by just 0.2 microns apart, which is, according to the Abbe's law, the maximum theoretical resolution on a visible light microscope. And this is something that fascinates me, how they could do it, how could they predicted this such a long time ago, and on the primordium of the light microscopes. In practice nowadays we can actually go a bit further in resolution, but some tricks have to be used, some mathematical tricks or other systems that it's not on the scope of this talk. But by using a light microscope, this resolution cannot be broken by direct observation. The important thing was not everything was yet perfect, although we could have that power of resolution. Lenses had to be improved, and they continued to be improved for a long time. And especially in order to correct the different aberrations, chromatic aberration, spherical aberration, and depending on the color that we were looking at, we would have different problems. So I told you before, so we already had the description of the chromatic lenses that corrected the blue and red. But then Autoshot was the first one to describe lenses that are optimized to correct blue, green, and red lights. So it's capable of correcting the three different wavelengths and focusing them all at the same point. As you can see, the lens starts to be complex, and they have to be perfectly adjusted, and constructing, manufacturing lenses without any aberration was an art. But nowadays, when you look into the lens of a microscope, they will have written something like this, depending on the scope you're using. They will have apochromatic, plan apochromatic. They will have different names according to the corrections that the lens is capable of doing. In fact, modern lenses are incredibly complex on the inside, and they have several movable parts to correct different types of aberration. So a single planapo lens, as shown here, can have as many as 11 lens elements on the inside. So there are multiple types of lenses with different elements, each one designed for specific applications, and they do different type of corrections. I've included this table for you to appreciate that the achromat type of lens, it corrects spherical aberration for one color and chromatic aberrations for two colors, and you can see the plan acro. It's capable of doing the same thing, but it corrects also the field curvature that I had no time yet to mention to you. And of course, the lenses that are the most expensive and do the most corrections are the plan apochromatic that you see in here. They're capable of correcting 3-4 colors, both spherical aberration and also in the chromatic aberrations. But lenses are incredibly complex, but you will have another seminar, another class that will cover this topic. Just to remind that these different aberrations that light can suffer when go through glass, they were all described by the beginning of the late 19th century, and they've been trying to be corrected by the manufacturers ever since. One of the problems that light can create to us, to the observer, is because the wave, they don't all are not incident on the sample all with the same angle of rotation, like you can see in here. They can come in different types of rotations, and Cernamont also in the 9th century was capable of appreciating that problem and created a polarizer that basically is capable of cutting most wavelengths except the ones that all that come in the same direction. This is better seen in this slide. So just to try to explain what I'm trying to say. So in here, let's imagine that we have light rays that come only from two different directions, one in vertical represented in orange and one in blue represented and one horizontal coming represented in blue. And the polarizer filter, basically what it does, it only allows the wavelengths to pass through these slots, and the blue rays are prevented from passing. So all the light that passes from the polarizer, the wavelength, it's all in the same direction. Is this so important? In fact, it is. So we have here an image taken on a pool, and in the same image taken without polarizer and with polarizer is completely different, in the sense that the degree of detail we can get with the polarizer, it's much higher than without polarizer. So of course this is due to the characteristics I've told you before: the reflection, the incident of the angle, and the refraction of light. So with the polarizer we can filter most of the undesired and desired angles of light, and we can just focus on one single type of light being reflected from the pool, and it allows a better resolution of all the details. In the beginning of the 19th century, William Wollaston made a huge contribution to optical microscopy when he created the Wollaston prism, that basically is an optical device that manipulates polarized light and separates the light into separate outgoing beams that will be polarized according to the optical axis of the right angle of the prisms. This sounds a bit complicated, although it's represented, it's simplified in here. And the main thing is the outgoing light. Will the beams diverge as two rays that will passage through the sample at very close adjacent points, around 0.2, and because of this they will experience different optical path lengths because of the refractive index or thickness of the sample at the site, and it will also cause a change of phase in one of the rays relative to the other due to the delay experienced by the wave in the more optical dense material. How can we visualize this? In order to help you visualize, I will also take advantage of the contribution made by George Nomarski that developed further the Wollaston prism, and the contribution of these two individuals nowadays is what we call the differential interference contrast. And as you can see, to visualize what's happening is a transparent sample that otherwise have no contrast, like in here in the bright field, with the DIC or the differential interference contrast, we can enhance the contrast of on a sample that it's otherwise not colored, no contrast added to it. And so basically we can obtain this image because of the splitted polarized light into the two beams that we can further see in this representation. So the incident light, the beam is splitted in two rays, it will focus into the two adjacent points, and as soon as there a difference in the material, like for example a membrane that it's very close, so one of the beams can pass through the membrane, the other one can pass to the medium immediately adjacent to the membrane, and this is what allows us to obtain those images without using contrast or chromophores or any other stain to give color to our sample. So basically in this presentation what I've been telling you is how microscopes were developed in the beginning, but just a simple microscope. So I didn't told you about immunofluorescence or data techniques. We just talked about bright field microscopy, where you don't use any stains, we just illuminate the samples, and it's very difficult through bright field to obtain structural details because usually the samples lack contrast. Anyway, knowing these characteristics of the light, we can observe these structural details if we manipulate the light in such a way that we can see phase differences, and we could further do it if we stain the components. But the main effects that we used of the light, so diffraction, refraction, reflection, can actually produce quite a good level of contrast and detail in our samples. So furthermore, we should have a good notion of all these characteristics of light. This is just a summary of the characteristics I was using today of the light. There's more that we have no time to talk about, but just by using the absorption of light, refraction, diffraction, and dispersion, we can have some very good detailing on our samples. On the other hand, I also mentioned you that there's a limit to what we can resolve, to what we can see just by using direct light in our sample. So I'm referring obviously to Abbe's law that I talked a bit before. That doesn't mean that we cannot magnify our samples more and more, either by using mathematical tricks, let us say, but the oldest way to have a higher magnification was, instead of changing the light, we cannot see it, we can use waves that have a very small wavelength, like a beam of electrons. So in 1927, Louis de Broglie invented basically the electron microscope, because electrons, like photons, can behave like waves. And there's the electron microscope that has the cannon of electrons, and we have a schematic here on the right of the slide. So this is actually the scheme of an optical microscope, where we have a light source, we have lenses, we have our sample, we have further lenses to amplify the image, and then a detector, or it could be our own eyes. An electron microscope, instead of having a light source, it has an electron beam that will send the electrons through our sample. Instead of lens made of glass, we have magnetic lens that will focus the beam on the sample, and then the beam will be detected by a sensor, a camera, a fluorescent plate, but never with our own eyes. So this is actually the limitation of the electron microscope. Although it can amplify, it can magnify, as you can see here, much more than an optical device, but we cannot actually observe directly the sample with our eyes, and most important of all, because of the high energy of the beam, the samples are never alive, so we cannot look into live samples, and the samples must be fixed. So in order to have a high resolution image, like the one we see in here, a photo taken with an electron microscope, to have such a high degree of detail, we have to sacrifice something, and that something usually is we never observe live samples. So I hope you have enjoyed this small presentation with some basic details of the optics that we use to build simple microscopes, along with a little bit of the history of microscopy.